New Nature Cancer papers reveal that total intratumoral bacterial burden, not PD-L1 or tumor mutational burden, predicts failure of immune checkpoint blockade in head and neck cancer. Here's what the data shows, why it matters clinically, and how this paper sets a benchmark for clear mechanistic cancer biology. I'm Dr. Susan Heavey. Welcome to Further Reading. In this video, we're walking through a paper by Silver and colleagues that explores the role of intratumoral bacteria in cancer. We'll look at how bacteria are detected inside tumours, how they interact with cancer and immune cells, and why this challenges the way we think about tumour biology, treatment response, and resistance. This isn't a microbiome overview, it's a mechanistic, data-driven paper that sits at the intersection of cancer biology, immunology, and host microbiome interactions. I thought it was a super cool read, and I'm looking forward to talking it through with you. I'm going to start with the panel-by-panel discussion, as always, before looking at the big picture. You can use the chapters to jump around the video if you like. So Figure 1 asks why some head and neck cancers respond to immune checkpoint blockade while others don't. Panels A through E clear the ground. Clinical features, PD-L1 expression, tumor mutational burden, baseline immune cell populations, and HPV viral load do not distinguish responders from non-responders. So none of the usual biomarkers explain response in this cohort. Panels F and G introduce tumor bacterial burden, the total amount of bacteria within the tumor. Here, the signal is clear. Tumors that respond to immunotherapy have significantly lower intratumoral bacterial burden, whether response is defined radiographically or pathologically. This is not about microbial diversity. Panel H shows no difference in Shannon diversity between responders and non-responders. Panels I through K address whether this effect is driven by specific bacteria. Although Fusobacterium correlates with bacterial burden, multivariable analysis shows that only total bacterial burden independently predicts immunotherapy response. So, the key message from Figure 1 is simple. In head and neck squamous cell carcinoma, resistance to immune checkpoint blockade tracks with the amount of bacteria in the tumor, not PD-1, not mutational burden, and not immune cell composition. Figure 2 asks whether tumor bacterial burden holds up beyond the original Chow cohort. Panels A and B start with an independent head and neck cancer cohort treated with immunotherapy. When tumors are stratified by tumor bacterial burden, higher burden is associated with significantly worse progression-free survival. In contrast, stratifying by Fusobacterium abundance alone shows no association with outcome. Panels C through E then move to an external non-small cell lung cancer cohort. Here, tumor bacterial burden again predicts immunotherapy response, outperforming both Fusobacterium and Escherichia. This is clear in the regression analysis and reinforced by the ROC curves. Tumor bacterial burden has the highest predictive accuracy. Panels F and G broaden this further across multiple cancer types. In multivariable models, tumor bacterial burden remains associated with response to immune checkpoint blockade across cancers. Importantly, this association disappears when patients receive chemotherapy or targeted therapy, indicating specificity to immunotherapy. So, Figure 2 validates tumor bacterial burden as a robust cross-cohort predictor of resistance to immune checkpoint blockade, and shows that its effect is immunotherapy specific. Figure 3 asks what tumor bacterial burden is actually associated with biologically. Panels A through D focus on the microbial landscape. As bacterial burden increases, microbial diversity falls, and the relative and absolute abundance of multiple tumor-associated genera rise, including but not limited to Fusobacterium. Crucially, tumor bacterial burden correlates with the total abundance of many bacterial genera, not just one species. Panels E through G then examine clinical correlates. Tumor bacterial burden does not vary with tumor size or stage, but it does differ by anatomical site and HPV status, with higher burden in oral cavity tumors and lower burden in HPV-positive disease. Panels H through J move into tumor biology. Higher bacterial burden is associated with specific genomic alterations, including Notch pathway mutations, and with increased expression of inflammatory and immunomodulatory proteins. Gene set enrichment analysis shows activation of inflammatory signaling pathways, including TNF and interferon alpha responses. So, Figure 3 establishes that tumor bacterial burden reflects a broader biological state, linked to tumor genetics, inflammatory signaling, and anatomical context, rather than simple contamination or microbial composition. Figure 4 asks how tumor bacterial burden reshapes the tumor immune microenvironment. Panels A through C show a consistent pattern across datasets: TCGA whole genome sequencing, independent exome samples, and the Chow trial. As tumor bacterial burden increases, neutrophils increase, while adaptive immune cells, particularly T cells and B cells, decrease. Panels D and E summarize this shift using the intratumoral neutrophil to lymphocyte ratio. Higher bacterial burden is associated with a higher TNLR, and higher TNLR, in turn, is associated with worse immunotherapy response. Panel F shows this relationship is not unique to head and neck cancer. Across multiple cancer types, tumor bacterial burden consistently correlates with an elevated neutrophil to lymphocyte ratio. Panels G through I provide orthogonal validation using imaging. Direct visualization confirms that tumors with more bacteria have fewer T cells, more neutrophils, and a markedly higher neutrophil to T cell ratio. Panels J and K then point to mechanism. Exposure of tumor cells to intratumoral bacteria induces conserved expression of neutrophil recruiting chemokines, mirroring gene expression patterns seen in high-burden patient tumors. So, Figure 4 shows that high tumor bacterial burden is linked to a neutrophil-dominated, immunosuppressive microenvironment, providing a mechanistic bridge between bacterial burden and resistance to immune checkpoint blockade. Figure 5 moves from association to causality. Panel A outlines the experimental design. Head and neck tumors are implanted either orthotopically in the tongue, where intratumoral bacteria naturally accumulate, or subcutaneously, where bacterial burden is low. Panels B and C confirm the model. Orthotopic tumors contain abundant intratumoral bacteria, and this bacterial burden is effectively depleted by broad-spectrum antibiotics. Panels D through G show the functional consequence. Antibiotic treatment significantly slows tumor growth in orthotopic bacteria-rich tumors, but has no effect on subcutaneous tumors with low bacterial burden. Panels H and I explain why. In orthotopic tumors, bacterial depletion reduces neutrophils and increases CD8 T cells, whereas subcutaneous tumors show the opposite or no meaningful immune shift. Panel J reinforces this site-specific effect at the cytokine level. Panels K and L then identify the key effector cell. Depleting CD4 and CD8 T cells does not eliminate the tumor suppressive effect of antibiotics. In contrast, depleting neutrophils completely abolishes it. So Figure 5 demonstrates that intratumoral bacteria actively promote tumor growth by sustaining a neutrophil-driven, immunosuppressive environment, and that by removing these bacteria reverses this effect in bacteria-rich tumors. Figure 6 completes the causal story by asking whether increasing intratumoral bacteria is sufficient to induce immunotherapy resistance. Panel A outlines the experiment. Mice receive oral inoculation with tumor-associated bacteria before tumor implantation, followed by anti-PD-L1 treatment. Panels B through F confirm the biological effect. Introducing bacteria increases intratumoral bacteria load, drives neutrophil accumulation, reduces T cell infiltration, and markedly increases the neutrophil to T cell ratio. Panels G through J show the therapeutic consequence. Controlled tumors respond robustly to anti-PD-L1 therapy. In contrast, tumors exposed to Fusobacterium, Prevotella, or Campylobacter become resistant to immune checkpoint blockade. Panels K and L rule out a gut-mediated effect. When tumors are implanted subcutaneously, where bacteria do not accumulate intratumorally, bacterial exposure no longer affects immunotherapy response. So Figure 6 demonstrates that increasing intratumoral bacterial burden is sufficient to induce resistance to immune checkpoint blockade, independent of bacterial species and dependent on tumor and local tumor colonization. So let's put this paper in context. Why does it matter? For the last decade, efforts to predict response to immune checkpoint blockade have focused on tumor intrinsic features, PD-L1 expression, tumor mutational burden, and baseline immune infiltration. But in head and neck squamous cell carcinoma, those biomarkers have repeatedly failed to explain why most patients do not respond. This Nature Cancer paper by Silver and colleagues reframes the problem. Instead of asking which immune markers are present, it asks a simpler question: how much bacteria is inside the tumor? Briefly, let's discuss the core contribution of this paper. So, using samples from what I'm pronouncing as the CHOW neoadjuvant immunotherapy trial, sincere apologies if I'm supposed to be saying CIAO, but I'm saying CHOW, the authors show that total intratumoral bacterial burden, not PD-L1, not mutational burden, not immune cell composition, is the strongest predictor of response to immune checkpoint blockade. Tumors with high bacterial burden are resistant to immunotherapy. Crucially, this effect is not driven by specific bacterial species. It's the total amount of bacteria, the biomass, that matters. And the biology is consistent. High bacterial burden is associated with neutrophil accumulation, loss of T cells, and an immunosuppressive tumor microenvironment. Most importantly, the authors go beyond association. By experimentally increasing or depleting intratumoral bacteria in orthotopic mouse models, they show that bacterial burden is sufficient to induce or reverse resistance to anti-PD-L1 therapy. That establishes causality. Now, this work does not stand alone. A companion Nature Cancer paper led by Riaz and colleagues analyzes the phase 3 Javelin Head and Neck 100 trial. In that randomized setting, high intratumoral bacterial burden again identifies tumors with increased neutrophil activity, suppressed adaptive immunity, and worse outcomes specifically with immunotherapy, but not with chemoradiotherapy alone. Together, these two papers provide rare alignment between mechanistic biology and phase 3 trial data. These findings also build on earlier spatial and single-cell work published in Nature. Nino and colleagues showed that intratumoral bacteria occupy organized microniches that are poorly vascularized, highly immunosuppressive, and enriched for myeloid cells. That spatial architecture helps explain why bacterial burden so consistently tracks with neutrophil-dominated immune suppression. At the pan-cancer level, a large cell study by Battaglia and colleagues confirms that microbial DNA is detectable across thousands of metastatic tumor biopsies. Across cancer types, microbial features correlate with neutrophil infiltration and immune checkpoint outcomes, although which feature matter most appears to be more tumor-specific. In head and neck cancer, this Nature Cancer work shows that total bacterial burden, rather than individual taxa, is the dominant signal. So why is this paper important? Well, taken together, this study shifts the field in 3 important ways. First, it reframes the tumor microbiome from a taxonomic problem to a quantitative one. Second, it identifies intratumoral bacterial burden as a clinically relevant, potentially actionable biomarker in head and neck cancer. And third, it provides a mechanistic explanation for why immune checkpoint blockade fails in many patients despite favorable conventional biomarkers. Rather than being immune cold, these tumors may be actively immunosuppressed by the bacteria they contain. This work places intratumoral bacteria alongside immune cells and cancer cells as a core component of the tumor ecosystem and forces us to rethink how we stratify and potentially treat patients receiving immunotherapy. One of the most valuable things someone new to paper writing can take from this work is not just the biology, but how the story is told. This paper starts with a very precise question, not what predicts immunotherapy response, but why do existing biomarkers fail in head and neck cancer? That clarity shapes everything that follows. Start with a precise question. The authors define one problem and commit to it. They don't try to solve immunotherapy resistance in general. They ask whether something fundamentally overlooked could explain it. That restraint is what makes the paper feel focused rather than crowded. Remove noise early. A key writing lesson here is how how quickly these authors eliminate alternatives. PD-L1, tumor mutational burden, immune infiltration, viral load, all are tested and shown not to explain response straight away. This clears conceptual space for the reader. By the time the central result appears, the reader already understands why it matters. Build one central axis. The entire paper revolves around a single variable, total intratumoral bacterial burden. Every dataset, every cohort, every experiment interrogates that same axis. There are no side quests here. This repetition isn't redundancy, it's reinforcement. Let the figures tell the story. Each figure makes one clear point. Figure 1 establishes the signal. Figure 2 validates it across cohorts. Later figures explain mechanism and causality. You never have to guess what the takeaway is. It's visually obvious. Use plain, disciplined language. Finally, the writing is deliberately simple. Claims are narrow. Verbs are concrete. The authors don't oversell clinical implications. They state what the data do and do not support. That restraint is exactly why the conclusions are convincing. The big lesson here is that clarity is not about dumbing science down, it's about deciding what the paper is truly about and then having the discipline to make everything serve that story. To summarise, this Nature Cancer work shows that resistance to immune checkpoint blockade in head and neck cancer is not explained by the biomarkers we usually rely on. Instead, total intratumoral bacterial burden emerges as a dominant driver, reshaping the tumor microenvironment, promoting neutrophil-mediated immunosuppression, and predicting poor response to immunotherapy. By combining clinical cohorts, randomized trial data, and causal animal models, these studies place intratumoral bacteria alongside immune cells and cancer cells as a core component of the tumor ecosystem. Just as importantly, they set a high bar for clarity in how a scientific question is framed, tested, and communicated. If you found this helpful, this video is part of a playlist called Cancer Biology and Oncology Papers Explained, where I break down high-impact cancer papers with a focus on mechanism, clinical meaning, and how to read and write clear scientific work. You'll find the full playlist linked here, and I'll see you next time.